Thanks very much, Elena. Thank you very much for your attention. And I would like first to thank a lot the organizers to invite me to this great meeting. So today I'd like to give a bit of a microbial perspective on insects. And you will see my talk will be mostly focused on microbes, which I believe are also super interesting organisms. But you will see that in our studies we use one very powerful insect, Drosophila, we heard about it yesterday, to study the mechanisms underlying the interaction between microbes, bacteria, commensal bacteria, and insects in the laboratory, to understand how benefits conferred by microbes to the physiology of insects are transduced mechanistically speaking. So biologically, I mean, the biological question that is under the focus in my lab is to study the physiology of animals, insects in particular. And we are very much interested by studying one physiological parameter, which is growth, juvenile growth and maturation. And that is extremely related to the microbial environment and the nutritional environment. And we are very much interested by understanding how those environments interact and impact the animal physiology. We apply a particular nutritional environment to the animals, which is chronic undernutrition. We do so because chronic undernutrition in human and animals is actually a condition that triggers stunting, which is a devastating condition that leads to reduced linear growth coupled to immune and neurocognitive development. And this is a disease that is extremely prevalent in human population, in kids in Africa and South Asia. Nowadays, with more than 150 million kids affected by those conditions, way more than obesity. So in this context, in this biological and application context, we do very basic science, basic research. We want to understand the mechanisms behind those phenomena. And we use, of course, model systems. And in the lab, of course, we use insects, Drosophila, and this is why I'm here today. But we try also to translate some of those results to mammalian models such as mouse. And we use a particular type of approaches using those animals, which is called notobiology, which is the discipline of breeding and studying animals in a microbial confined environment where you completely control the microbial environment of the animal. So for that, we associate axenic, germ-free animals with pure culture of microbes, and in our case, commensal microbes. So I don't have to come back to the great tools that we can use in flies to study mechanisms. I mean, we had a stellar example in the first talk in this conference. But what is very interesting with Drosophila, at least, is that it's very easy to generate germ-free animals and to breed germ-free animals in the laboratory. Coupled to that, the very interesting parameter also is that the microbial community associated to flies, at least in the laboratory, is extremely simple. In our lab, for example, we flies face two major bacterial species, Enterococcus fecalis and Lactobacillus plantarum. The communities in the wild are a bit more complex than those two species. And as I said, the life cycle is short in the lab, and we can really control the nutrition of those animals and the microbial environment by doing those association experiments, breeding germ-free animals and reassociating with known culture and purified culture of those particular microbes. So I was very much inspired by the work of Pierre Leopold in the past years in the fly field, where he showed that actually optimal nutrition lead to a developmental timing of 10 days, so from an egg to an adult. And he previously showed that chronic undernutrition was really a phenomena that impact both developmental timing and the size of the animal. I'm sorry, the picture doesn't show up here. But the impact, the phenotype of stunting is really clearly observed also using insects during chronic undernutrition. And Pierre's lab and other labs in the world have dissected out some of the mechanisms, hormonal and genetic control mechanisms that control those phenotypes. In this context, we were very intrigued by what is the contribution beyond chronic undernutrition, what is the contribution of microbes in this context. So if you breed germ-free animals on the normal diet, you get adults in about 10 days. Very similarly, that animals with microbes, there is a slight delay. But when you undernourish, you chronically undernourish the animals by reducing the yeast component of their fly food, you have a 5-day delay that can be compensated, at least partly, by the addition of some bacterial commensal strains, purified strains, to the media associated to the flies. And that is not only reduced in developmental timing, but also you see that the size of the animal, the growth rate of the animal is boosted very early during larval development. So here you see the size of the larvae over time, and animals which are associated with some strains of this Lactobacillus plantarum species do grow faster than their siblings which didn't face the bacteria. So in this context, we've been dissecting the molecular mechanisms underlying that phenotype, and we published that over the last few years, and I will sum that up in this slide. So we first showed that peptidoglycan sensing, so peptidoglycan is a component of the cell wall of bacteria, and gram-positive bacteria, for example, as for Lactobacillus plantarum. Peptidoglycan sensing triggers innate immune sensing, innate immune response in the gut, but for lactobacilli and commensal bacteria, actually it's not mainly immune genes which are induced, but more rather digestive enzymes which are induced by this signaling pathway in enterocytes in the gut when peptidoglycan is sensed. And those host peptidases are induced transcriptionally. There is an increase in peptidase activity in the intestine, which is optimizing the digestion of dietary protein and the assimilation of dietary amino acids. And those circulating amino acids are known signals, and again, I refer to the work of Pierre Leopold that has shown that those amino acids are really inducing systemic signals such as growth factors and growth hormones, which triggers this increase in growth rate and entry into metamorphosis, so maturation steps. So here, by sensing animals in the intestine, optimizing digestion and triggering this endocrinal signal, we get a systemic phenotype thanks to the presence of a single bacterial isolate in the diet. So that phenotype is extremely quantitative. We can quantify it very simply by associating germ-free animals with pure culture of different strains, let the animal develop on the poor diet, and just study the size of the animal a week after the treatment. So you see it's a phenotype that is extremely robust, and it gives us a phenotypical window in which you can screen for host or bacterial genes, which are at the basis of that phenotype. So before screening, what we did is looking at different bacterial strains and look at the impact of those bacterial strains on larval growth. And as you can see on this graph, we could separate strains as super growth promoter, intermediate growth promoter, or non-growth promoting strains. All those three categories, the bacteria grows very well on the diet and associates very well with the larvae. So there is no difference in colonization. The red ones are actually, so when the larvae is similar in size than the germ-free, relates to the fact that those strains were not able to colonize either the animals or the diet. So we have a very strain-specific phenomenon, and we were very intrigued by understanding the genetic basis of this strain specificity. So we took a step back and we decided to screen genetically the genome of Lactobacillus plantarum in this context. And for that, Renata Matos, at that time a postdoc in the lab, has been designing a transposon-mediated random mutagenesis of the plantarum genome. She generated a library of 2,000 random insertions and screened them for loss of function of this growth promotion phenotype in flies. And after some weeks of screenings, she focused on 7 candidates, and particularly one insertion in this peculiar operon, which is the PBPX2 DLT operon. And that operon is actually encoding for a machinery that is known to have alanines on teichoic acids, and teichoic acids are another component of the cell wall of gram-positive bacteria. Knowing that peptidoglycan sensing is very important for the phenotype we observe, we are very intrigued that we recover a gene that affects another structure of the cell wall in our screen, so we decided to focus on that gene. So what Renata did first is to confirm the insertional mutant phenotype by generating a clean knockout of the gene which is impacted by the insertion. or deleting the 3' genes in the same operand or the whole operand. And as you can see on this diagram, we look at the size of the larvae as a germ-free or associated with Lactobacillus plantarum, the wild-type strain. And you can see the initial mutant, insertional mutant phenotype, the growth promotion is affected. The larvae are small when they are associated with this mutant. And we recapitulated the mutant phenotype only by deleting the entire operand. So this is clearly all the genes of this machinery which are required to mediate the phenotype. So what was known, as I said, is that this peptidoglycan sensing triggers a host peptidase activity in the gut of the growing larvae. So we went on analyzing in larvae which are either germ-free or associated with the wild-type bacterial strain, the proteolytic activity of the intestine in the lumen, in the gut of the intestine, and we could see, as we previously showed, the increase in activity. But that increase of activity was decreased when we used the mutant strain. Interestingly, when we look at the expression of the genes which encode the enzymes responsible for this activity, we could see a decrease. But that decrease was partial, most likely because this pathway, which is based on peptidoglycan sensing, is still fully active. So we went on testing that directly by repeating those experiments in a mutant fly that has an IMD pathway completely blocked, a loss of function mutant in the DREAD gene, which is a gene which is essential for that pathway to function. And we repeated the expression phenotyping of the animals. I recall you the wild-type situation, a partial reduction and peptidase induction. And now, as you can see, in the mutant associated, the mutant bacteria is associated to animals, we don't see anymore an induction of the peptidase, suggesting that there is indeed another mechanisms beyond peptidoglycan sensing that is required to trigger peptidase, and that is dependent on that feature which is controlled by the mutant. We know that this operand is actually decorating the teichoic acid, so we were proposing that actually this is two ways of recognition. So beyond host peptidase expression, we went on looking at larval growth, and we could see an additive effect of being mutant for the cell wall machinery, the teichoic acid decoration machinery, and being mutant for the IMD pathway, also for the larval size phenotype, so the macroscopic growth phenotype. So we propose that both peptidoglycan and additional signals, such as the dealanylation of teichoic acids, are required for optimal plantarum-mediated growth promotion in this system. What was very important to test was whether it was a direct sensing of the cell wall that is required to trigger that phenotype. So for that, we purified, thanks to a collaboration with Marie-Pierre Chapeau-Chartier at INRA in Rouy-en-Josas near Paris, she purified with her team cell wall from either the wild-type bacteria or the mutant bacteria. And we treated germ-free animals with those purified cell walls. And that was not enough to trigger a growth phenotype. We did the same on the animals associated with the wild-type bacteria, and the same, we couldn't see any difference. What was very interesting is that when we took animals associated with the mutant bacteria, then we could increase their growth-promoting phenotype by adding the wild-type cell wall, but not the mutant cell wall. So that was a clear indication that there is a direct cell wall sensing involved into the phenotype and other additional signals that we have not yet fully understood. So that was a story we published last year, which started to establish some of the genetic basis of plantarum-mediated growth promotion. And here on this summary slide of that part, we can see now that there is most likely two sensing mechanisms that trigger this digestive enzyme response, which contribute to systemic growth promotion. I would like just quickly to mention another story we published this year, well, actually last year, that established that this Plantarum Drosophila system is really a case of mutualism with a mutual benefit. So larvae benefit from the presence of the commensal partner, Lactobacillus plantarum, and which trigger growth. But also the bacteria benefit from the presence of the growing larvae in the nutritional niche, because the growing larvae is shedding a lot of sugars in their excreta. And those modified sugars, N-acetyl-glucosamine, for example, are actually extremely good carbon source for the bacteria. So the bacteria also, the titer, maintain over time, thanks to those sugars which are shed by the larvae. So it's really a bidirectional benefit. So coming back to the second part of my talk, coming back to this interesting observation that we have a strain-dependent phenotype, another postdoc in the lab, Maria Elena Martino, was very much interested to see whether she could evolve a non or mid-growth promoting strains into a super promoting strains, thanks to bacterial evolution and experimental bacterial evolution. So to do so, what she did is to design a protocol over 20 fly generation, where she would monoassociate germ-free fly eggs with an ancestral record, an ancestral strain, a non-growth promoting strain or poorly growth promoting strain, which is the ancestral strain we use in this protocol. And then she would wait for the first pupae to emerge, select the first 10 pupae out of a cohort of 40 eggs that she associated, and transfer in a sterile environment those pupae which are associated with microbes in a new tube, wait for adults, let the adults mate and lay new eggs, and do so on and so forth for 20 fly generation. So this imposed a very strong bottleneck in selection pressure, and we were expecting to select for de novo mutation and to select for clonal interference by indirect selection of the most beneficial bacterial strain, because bacteria divide way more. There will be way more bacterial generation than fly generation in this system. So first, we were very happy to see very quickly that actually when we started the experiment, first generation, it's about 12 to 13 days between the eggs to the adult stage. And then very quickly, after a few fly generation in the protocol, we had new flies emerging very quickly to pupae emergence in about a week. So we had already acceleration in the system. So that was a very good sign. So what Maria Elena did is to sample at each fly generation many isolates and test them one by one on a new batch of germ-free animals, naive germ-free animals, and she tested their ability of all those strains to promote growth and compare the ability of those evolved strains to the ancestral strain or the actually growth-promoting strain, the WGL strain. And she quickly realized that she started to get isolates which behave like the WGL strain, while in fact they are derived from the ancestor strain, which is not growth promoting. So that was super exciting, and she realized also that very quickly, most of the isolates, as you can see on this slide with the color code, most of the isolates were actually more beneficial than the ancestral strain. So evolution had occurred, and that was cool. So then she repeated that, exactly the same setup, a few months after, exactly the same systems, and evolution occurred again, as expected. So she got again an acceleration of the phenotype. She got again a lot of isolates which showed an improved growth promotion capacity as compared to the ancestral strain. And again, the incidence of those isolates in the population was total in few fly generation. So obviously, she went back on sequencing the genome of all those or many of those beneficial strains just to see what are the mutations, is there mutations, and what are the mutations that were accumulated in the genome. And for the first experiment, she found that after 2 fly generation, she already got this allele in the ACK gene, and then other mutations showed up in the protocol. So this is generation 2 of the fly up to generation 20. And what was super exciting is that in the second experiment, we got other mutations, but again, the same ACK gene that was mutated. So the interesting fact is this mutation for the second experiment is actually a stop codon emerging very early in the protein, while in the other experiment, it's a single triplet nucleotide deletion, 1 amino acid deletion. That seems to behave similarly than a loss of function genetic mutation. And having strains with only that mutation that are already growth promoting as compared to the ancestral strain was a very strong indication that mutation was actually causal to the phenotype. But to be fully sure, we collaborated with a lab that masters very well CRISPR-Cas9 strategies in lactobacilli, which is not as trivial as you would expect. And what the lab of Chase Basil could do is to actually engineer the evolved strain, which has lost the 3 nucleotides in the ACK genes, and engineer reinsert back the triplets in the genome of that strain. And we checked the phenotype of that strain. And what was interesting is that reverting this single mutation actually reverted to phenotype back to the ancestral strain phenotype. So that was a very important demonstration that evolution was, phenotypical evolution was actually caused by that very specific mutation in ACKA. ACKA is actually acetate kinase gene, one of the two acetate kinase genes, which is therefore core to the metabolism of Lactobacillus plantarum and acetate metabolism. So we went and studied the metabolism of this evolved strain as compared to the ancestral strain. And what was extremely clear from the metabolomic profiling in this context is that there is a strong accumulation in the supernatant of those cultures of N-acetylated amino acids. So those strains, those evolved strains, release a lot of N-acetylated amino acids. And for now, we have no clue what's the connection between the mutation and that production. So the very simple experiment we did, and that is the last experiment I'll show you, is we complemented either germ-free animals or animals which are associated with the ancestral plantarum strain or animals associated with the evolved strain with increasing concentration of one of the most representative N-acetylated amino acids, N-acetyl-glutamine. And N-acetyl-glutamine could only promote growth to a very high, when it's provided at very high concentration to germ-free animals. But at lower concentration, it was sufficient to show a phenotype, to rescue the evolved phenotype as compared to the ancestral phenotype. So we could mimic the evolution by just providing the N-acetyl-glutamine, the evolved phenotype by just providing the N-acetyl-glutamine to the ancestral strain. So that actually established that beyond this cell wall sensing that I previously discussed and this digestive function that I previously mentioned, there is also a very strict and direct metabolic crosstalk. And we heard from Angela this morning that there is this metabolic crosstalk between symbionts and insects, that in this case, the bacteria has evolved to actually produce more N-acetylated amino acid, and in particular N-acetylated glutamine. And that provision of glutamine is actually extremely good for the animal to grow faster. So I hope that I've been able to show you that by using insects and Drosophila in particular, we can really go into the system of how a nutritional environment and microbes shape actually the response of the animals to this nutritional environment and to a very mechanistical basis. And I just mentioned that what we've done also in the past few years is to translate some of those discoveries in mammalian system, in murine models, by associating germ-free mice with different strains, and actually the strains that promote growth in insects. And we were extremely pleased and surprised to see this strain-specific growth promotion phenotype also using germ-free murine models, suggesting that this lactobacilli-mediated growth promotion is probably conserved during evolution. So with that, I would like to thank the people that did the work, and especially Renata Matos, who's now a researcher in my lab at CNRS, and Maria Elena Martino, who's now a PI in Padua University in Italy, and our collaborators. And I mentioned here also the key people that have been developing the fly work I've been presenting in the first slides, and the colleagues now in the lab working on the mouse projects, and the sponsors, and you for your attention. Thanks a lot.
Thank you, Francois. We have time for questions. Okay, I will start probably. So you just defined this factor which promotes larval growth. And do you know how it promotes larval growth in Drosophila? So what are you doing now probably to identify the signaling works behind it?
Yeah, so now we are coming back to using the genital toolkit of the flies to actually screen for potential receptors and signaling molecules expressed in enterocytes, which might be sensitive to this modification of teichoic acids. It's actually very exciting because, these peptidoglycan sensing system is conserved in mammals. There is the nod receptors, which are very important. There are also PGLPs in mammals. But the sensing of lipoteichoic acid has been extremely controversial. And even in mammals, they still don't know the lipoteichoic acid receptors. There have been recently some interesting work on whole teichoic acid, but not on lipoteichoic acids. And especially dealinylation of those teichoic acids. We don't know what's the host receptor to that. So we hope by doing some genetic screens in flies, we can progress in the knowledge of that system.
Other questions? Yes, Jan.
Yes, I have a question on your gnotomutic model with lactobacillus. Do you know if in this kind of experiment if the bacteria is destroyed and participate in the nutrition of the host? And if you induce just, for example, if you give peptidoglycan only and trigger the peptidase, etc.,
yep.
You have partial rescue of the phenotype. It's not the whole.
Yeah. So there is clearly a strong elimination of the bacteria when they go through the gut. So there is a contribution of the bacteria as a trophic source, as a food source, like in C. elegans, for example. They are eating microbes. But beyond that, I would say we quantify that in a previous paper, that it's about 20% of the phenotype can be explained by just providing matter and trophic source. Beyond that, there is all these biologically and genetically encoded mechanisms. Now, indeed, some of the bacteria are killed in the gut during the passage in this acid zone in the middle of the larval gut. When genetically we engineer flies not to have this acid zone, we don't have this killing. The whole population of bacteria passes through without being killed. And in this context, we still have a very good growth promotion. So it tells you that it's not down to just killing this bacteria and digesting it and get the biomass of it that explain the phenotype. I'm not ignoring the fact that there is a contribution, but there is a very strong biological system that actually explains also the strain specificity of that phenotype.
Well, Ian?
I take it that your experimental evolution studies were done with flies on low-protein diets.
Yes.
What would have happened had you used the high-protein diet?
We don't know. We've not done it, but actually you mentioned that, and if I didn't say it, that's a mistake, because this parameter it's key, because actually the bacteria didn't show you the whole paper, but the bacteria adapts to this poor diet. And that's adaptation to this poor diet that somehow select for the persistence of that variant in the context. We repeated that experiment without flies, just letting the bacteria on the poor food, and actually this ACK mutation popped up. But then, as a positive bystander effect, that mutation triggers this release of N-acetyl-glutamine and other amino acids, which is capitalized on by the larvae and makes the larvae grow better. So again, that's the bacteria that adapts to the poor food, but then the partner, the fly, capitalize on that and makes it better on the poor food. So it's really this association and a mutual benefit that is really biologically exciting and interesting in this system.
Well, last question.
So François, very nice talk. So there is this trophic membrane inside the gut, which normally contains the bacteria. So how do you think the cell wall sensing system is working?
It's a very, very interesting question. It's very well known that when bacteria divide and they shed pieces of peptidoglycan, and these teichoic acids, they are branched either on the lipids or on the peptidoglycan. So we believe that actually there is no need of having a direct contact between the apical phase of the cells, of the enterocytes, and the bacteria to trigger a signal, because all those molecules, all those compounds are actually circulating and diffusing. And we also believe that, and it has been shown by the lab of Bruno Lemaitre, that actually the peptidoglycan can be found in hemolymph, clearly in the fly in adults and probably in larvae also. So you see, this is a system where the molecules are diffusing.
Well, thank you very much.
Thanks a lot.
And